Hey everybody, this is John at the top of the episode. I wanted to do a quick disclaimer before we get started. The views represented here are our views and our views alone. They do not represent that of our employers. To that end, please do not try and find our employers either. Thanks.
Hi there, welcome to seedpod, podcast about the natural world. My name is Kelvin, and I will be the host for today. Before we jump into things, I just want to introduce my co-hosts.
I'm AJ.
And I'm John.
All right, so today we're going to be talking about chronic wasting disease. Before we get into chronic wasting disease, it's time for our first segment. It's the invasive species of the week. Woo, 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 woo.
Air horns, gamer horns go here.
Yeah, so this is our first mammalian invasive species. So today the invasive species is the cicadeer. Cervus.
And not Sitka, as I thought it was.
Yes, Sitka.
Correct. That's a different thing.
So cicadeer, Cervus nippon, also known as Japanese deer, as the specific epithet would suggest. So this is a deer that is native was formerly native to a large swath, to a good chunk of Asia, from Russia down to Vietnam, also into, and in a couple of places in Russia, a couple of places in China, and found commonly throughout. So it was native from Russia to China, southern China, and across Vietnam. Currently, it's mostly limited to its native range, Japan, and a couple of places in Russia and China. However, it has been widely introduced across the world for game purposes, as a hunted species. So it has been introduced, across Europe. I have actually seen this deer in Ireland, where it was introduced for game purposes. But where I'm most familiar with it is in Maryland. So it was introduced for hunting purposes in the early 1900s. And
Of course it was.
yeah, and actually the Maryland DNR manages it as a hunted species because it's kind of a unique thing to hunt. So it's not the largest deer. It gets up to about 90 pounds in Maryland. I think it can get a little bit bigger, but it's not a huge deer. And some of the Sika deer have spots on them still, in their old, even when they're mature. So that's kind of a unique thing about Sika deer. In Maryland, they find them in the swamps. I think it's on the Delmarva Peninsula, right? Kind of across from where DC is. So it's kind of an interesting little critter there. What's really interesting about Sika deer is they do grunting. They have this really interesting grunt. It's how you can kind of hear them and figure them out. But they're very damaging to native ecosystems. In the United States, obviously, they're a deer species. They're in cervid, which is why we're talking about them today. And so they eat just about everything they can get their faces on. Because Maryland is in the Eastern United States, they have no predators. Coyotes will prey on fawns, but it's not heavy predation, and they, pretty good at reproducing. But we can find them in a couple of other places. AJ, I think you had a fun interaction with one.
Yeah, we had one that escaped from somebody's, high fence operation and showed up in town. Like somebody spotted it behind a grocery store and called it in basically, and then we went and rehabilitated the shit out of it.
Rehabilitated.
Biggest euphemism ever.
It was rehabilitated to a permanent end.
That's exactly right. Rehabilitated to a permanent end.
So Sika deer, invasive in the United States, Europe, and a couple of places in Asia, largely introduced for hunting. This is an interesting, animal in that it's a great demonstration of the social side of invasive species. These animals are generals that do well in lots of places, and they wouldn't be there without hunting. And they're maintained in a lot of places just for hunting, even though a lot of, there are plenty of deer in Maryland. There's no reason to keep them, but they're kept, as a unique thing to hunt. So the other thing that's really interesting about Sika deer is some of the subspecies are extinct and endangered to extinct And so there's this is one of those times where you can have an endangered animal that is invasive. So in its home range, this animal is protected and important, but in its invaded range, it's a hugely problematic and should be removed, and should be removed from the ecosystem. So this is a really interesting thing. We're going to talk more about endangered animals that are invasive and kind of the interestingness that brings up, like tahr in Texas, and I think tahr are also in New Zealand. So Himalayan tahr, it's kind of like a goat.
Oh, okay.
Yeah. But that's a different animal for you. Yeah, sorry, John.
No. I have a question. Do we know why it was introduced?
Oh, tahr?
Everywhere? Or no. Sorry.
Just for hunting.
They're
Okay.
just for hunting.
Yeah.
So is it like a interesting thing to hunt, or is it, oops, we've hunted everything else to extinction in the area, let's just grab this stuff from Japan?
No,
cool.
it's interesting to hunt. Interesting to hunt.
Okay.
Yeah. The releases in like England and Europe were done for hunting purposes, just because this is a cool thing that we can hunt. Same with the U.S. We do also have to remember that there was especially in the 1800s, there was that Sinoism, where everything Japanese was really cool, and we kind of do all of that, and so.
Like the Oriental.
Yeah, that's the word. Yeah, that's it.
Or is it something else?
Yeah.
Okay.
So that was definitely one of the things, and then a lot of those 1800s, early 1900s hunters, and still today, people really like having different trophies and taxidermy things.
Yeah.
And not everyone can go to Japan and hunt sika deer, but if you've got sika deer in, let's say, Virginia at a high fence, you could charge a lot of money for some people to come there and shoot that. So that's why it was exported places.
Gotcha.
Yeah, and people want to pay money to go shoot it, so they do.
Do you know when most of the introductions happened in the U.S. at least?
1900s.
It was the 1900s.
Yeah,
okay.
early 1900s, and I'm sure that there are continued introductions from escapes.
Well, yeah.
I know that there. I'm seeing that there are potential releases and escapes in a lot of states, but the two that I'm aware of where it's definitely established are Texas and Maryland.
Yeah. Yeah, those are the big ones that I have at least heard about.
Yeah.
I think everywhere else is just kind of like occasional escapes, but not a known established breeding population.
We will have a whole episode on invasive mammals in Texas eventually.
Yeah.
In Texas?
Oh, it's a whole thing. We're not going to get into it now, but it's a whole thing. Yeah.
Okay. I'll take your word for it.
Yeah. Okay, so that was our invasive animal. Now on to our second segment. It's our Forest Minute. Woo-hoo!
Yay!
AJ. Positive.
What is your Forest Minute?
I don't have a good one for this past week because I didn't spend a whole lot of time in the forest because it's been fucking raining.
You guys have rain?
Yes. So much.
God. We were supposed to get like a half inch, and it's nothing. Just nothing.
We've had like 3 separate thunderstorms today.
Jesus Christ. I'm so jealous. It was supposed to be raining all day today, and it's just been nothing. So continue.
It's just it's like as soon as it dries out a little bit, that the humidity can drop, it rains again, and it goes back up to 100, and then it drops a little bit. It's great. Yeah, we're just steaming in the slop over here. No, I guess a good one. We just had one of our. It's not actually in the forest, but it was a good thing. One of our state prescribed fire council meetings was last week, and I went up to that, which is always a good time. So fire councils are kind of I don't know the best way to describe them. It's basically folks that are practitioners of prescribed fire, kind of organizing and coming together to, talk about issues we've all had, challenges going forward, kind of usually, kind of the long-term climate outlook for the coming year. Is it going to be a busy fire season, a bad fire season, a good one, whatever, lessons learned, that sort of thing. But overall, just a really good meeting, a lot of good, networking, both for agency folks, but also, there's a lot of like private landowners and, contractors and private practitioners as well. So yeah, that was last week. That was a good there's not a whole lot of all day meetings that I'm like, yeah, that was great and a good use of my time, but that is, one of them. So it's nice to do that.
Love that people come together and talk about things and hopefully make improvements.
Ideally, yeah.
Okay, John, what was your forest minute?
So I've got potential. So I'll do the easy one. Is I did a direct seeding of hardwoods now almost 2 years ago. And so for the uninitiated, what you can do at a big enough scale is collect thousands and thousands and thousands of acorns and walnuts and all the other seeds that naturally occur in a forest, and you can plant them in the ground. We've got a machine that's basically all it is. It's a little plow that digs into the soil a little bit, and we've got a set of pipes that follows behind, and you drop acorns and walnuts and hickory seeds and other stuff down this tube, puts it 3 inches under the soil, firms the soil up behind you, and however many months later, all the acorns and stuff like that start sprouting. And I just revisited the first one I kind of did at scale. It was 6 acres, and it's growing so well. We have just tons and tons. It was almost all oak and a couple other just kind of shrub species thrown in there with like various forms of dogwood. And it's growing so well. The red oaks are like 1.5 feet tall. The bur oaks and white oaks are doing their little thing. They're like 6 inches, which is pretty normal. And so it's just this really cool success story that I've been able to do. And there's a lot of points of failure for direct seeding compared to other forms of tree planting, and so I'm just happy it survived. I got to get it past deer browse next, but I've got plenty of survival to do it, and hopefully I have literally overwhelmed it because I did a couple quick plots of measuring some seedlings and doing some quick math, and I've got like 5,000 trees per acre. So, and a pretty active group of hunters around us. So, fingers crossed.
That's fantastic.
That's awesome.
Yeah. What about you, Calvin?
My forest minute is we went out to this high grade forest. It was the graded forest that was overrun with ferns. It was like the average basal area was like between 40 and 80, or 40 and 60. So technically, it's a woodlands. It's not really a woodlands.
Yeah, I was gonna say it's definitely not.
Yeah,
good.
And so the understory is all New York and hay-scented fern, and so there's nothing else in the understory. Well, there was a lot of Japanese barberry. So we had a lot of invasives and fern, and just a complete just field of green, but nothing was growing and living there. And so we treated that last year. We went back this year to do a follow-up, and we killed like 90+% of the ferns, and we didn't get any sprouts back. And so we got to get some, native seed, some native tree seedlings coming in. That was really cool. And then we saw 2 rattlesnakes at the property, which was a lot of fun as well. Yep.
That's impressive.
Yeah, we saw them first thing in the morning. So the snakes were just big sleepy guys, and they weren't at all expecting us. And they were, oh, hey, what's going on, dude? It was also nice that we had killed the fern because if we hadn't killed the fern, it would have been a bad time just running those dudes.
You would have found them in a different way, perhaps.
Yeah, it would have been like 58. It was like 57 acres of just solid fucking fern. So probably would have stepped on one of those. That was my forest minute. Okay, so without any further ado, let's jump into today's episode. So today we're going to be talking about chronic wasting disease. AJ, I know you know a lot about chronic wasting disease. John, what do you know about chronic wasting disease?
So I know it primarily affects deer. Like it tends to be found in like really high deer population areas. It may not.
Do you know what it is?
So it's misfolded proteins.
Okay. Fantastic.
Beyond that, nothing.
Okay, fantastic. Excellent. This is a great place to start. So we're going to start all the way at the beginning. Just like in The Princess Bride, go back to the beginning. Back to the beginning. So let's go back to the beginning.
Once upon a time.
Right. Well, when you get, when the job goes bad and you get lost, you got to go back to the beginning. So follow me. I'll be your Vincini. So in the beginning, you start with proteins. What is a protein? Let's think back to basic high school biology. What's a protein?
God.
It's a God. It's the
It's protein.
It's protein. It's basic building blocks of life. It's amino acids and I don't know, some other shit that folds up and does stuff.
Yeah, exactly. Yeah, it's the building block of life. It tells you how to build muscle. Tells you to build. It tells cells how to work. It's just like how we communicate stuff in our bodies. How our bodies communicate with itself. So that's what a protein is. It's a chemical. And how do we know how they work? How does a protein work?
How does a protein work?
Fuck if I know, man.
Don't they like fit over the top of other molecules and disassemble them or something like that?
Yes, kind of. Yeah, so like we have...
That's like an enzyme, which I think is a type of protein.
Yeah. So
okay.
We have some proteins are just like, they're made of various molecules and they work, in high school biology, they work based on their size, so how big they are and what they're made of. So that's what you learn in high school biology, at least what I learned in high school biology. But that's not all proteins are. So like the classic protein is just like a 2D model of like a bunch of like, carbon bonds with some other shit in there. Because that's like, that's how high school biology is. You make it simple, you start simple, and then you build up from there. I mean...
Right, yeah. You can't represent a protein with, a chemical formula.
Right.
Because the shape is the name of the game. The shape is important.
Oh, AJ, going right to our next thing. Right. So, this is how in a lot of Western countries we teach science is we lie to you. So you make something really simple, and then as you learn more about it, you go, oh, that was a lie. That's just like a foundational disagreement that I have with how science is taught in a lot of places. But yeah, I'm just going to show you guys some stuff so we can follow along. Okay, so we start with proteins as a two-dimensional thing, but proteins are actually three-dimensional. So they work based not just on their size and their makeup, but also their orientation and their shape. So what is this, John? What does this look like right here? Can you just describe this for the listener?
Yeah, so it looks... I'm looking at...
So we start with the 2D guy. It's a classic diagram, and then what's this guy?
So the 2D diagram is a bunch of hexagons, right? And the 3D one that I'm looking at is a bunch of corkscrews, basically. Like flat paper corkscrews. Like if you took a piece of paper and you made a little, I don't know, confetti type thing that's curled and long. That's really
Yep. Yep. I think it's.
I don't know what it's supposed to represent besides proteins, but it's that like Mobius strip looking.
Yeah. So this is myostatin. The protein we're looking at is myostatin. This is what builds your muscles. So, listener.
Oh, that's why there's a picture of a dude with muscles.
Yeah. Listener, you can't see this because this is an audio format. But I have a picture from Predator of Arnold shaking hands, the shaking hands meme. And so we got two big two big dudes flexing their biceps. Okay, so so you get big muscles when your proteins are working correctly. So everything is correctly oriented. Remember, you're doing this hundreds of thousands and millions of times. So if you have an issue with your proteins, so John, what has happened now?
I can't tell the difference.
So it's flipped over.
It flipped.
Yeah, so
it flipped.
The protein...
Okay, sure.
The shape is the same, the orientation is just different.
Oh,
right.
Okay. Sure, yeah.
So if you have an issue with your orientation, the protein doesn't work. Because again, we have to remember that proteins are more than just what they're like made of. It's just like origami. It's how they're folded. It's how they're put together, what they look like. If I give you a crane that's upside down, it doesn't look like an origami crane. Same with your proteins. So you can have lots of issues with a protein. It can be misfolded. So that's again weird. Instead of our crane having two wings, now our crane has one wing and a really long neck. Doesn't really look like a crane. Or it could be made of the wrong paper. So then if it has the wrong stuff in there, then it's made of the wrong paper and doesn't really work as a crane. So these are all issues that can occur when you're transcribing or making proteins. Because your body does this so many times, issues are eventually going to arise. So that happens most of the time with most proteins. It's not that big of a deal. Your body just throws it out and it's like, all right, coolio, it's fine. We got plans for that. So That's what happens most of the time. Sometimes it doesn't work, and that's when we get some diseases. So what we're going to be talking about today is prion disease. So what does a prion disease sound like?
Something from like some sort of science fiction movie.
So
honestly.
I think this is where most. They're also the prions. Prions, I don't give a shit. So I'm going to say both. It's fine. So prions or prions are these really important proteins that are found in all mammals. They're highly conserved among mammals. Just in primates, there was a great study that looked at the similarity of proteins between humans and 25 other primates, and we were found to have between 92 and 99% similarities between prion protein. So this is a highly conserved protein that's found in all mammals. You can actually make mammals without prion protein, and they don't really survive because prion protein is very important. So we think it does a lot of things.
It's pretty foundational.
Yeah, it does everything. So it's in your brain, it's in your lymph node, it's in all muscle tissue, it's in spinal cord. So it does a lot of different things in mammals, like repair muscles, move copper, and help with sleeping. It's a difficult thing to study because when you remove it from a mammal, the mammal dies. And they die not well. So there's a lot of bad things happen when you don't have prion protein. So this is an important foundational protein. So if you don't have functioning prion protein, you die. Simple as that. Simple as that. So you can have prion issues for a couple of reasons. You can have a genetic mutation that gives you a problem, like sleeping sickness. Did you guys ever see? So you can have sleeping sickness is a genetic mutation. There are some genetic conditions that can lead to prion issues. You can also just randomly develop prion issues. So when that happens,
Like is that a mutation? Or?
Yeah. So that's just it's just a random, it's like a random typo that occurs with your prion protein. It happens.
Gotcha.
It's the cause of death in 1 in a million cases, and so that's the fun thing about studying prion proteins is sometimes you just have problems. And it's just nothing you can do about it.
Just happens.
Yeah, no, it just happens. It's just kind of dead. That's it. Because when your prion protein doesn't work, you die. So sometimes shit just happens. Oh, the other fun thing I forgot to say about...
fun thing, sorry.
The other fun thing I forgot to mention about prion is it's a shortened form of 2 words, proteinaceous infectious particle, because the term prion was come up with, after we figured out that there was this disease, and then we figured out what prion protein was, because this was discovered before proteins were actually nailed down as a thing. So that's what.
It's also a bird.
Oh, really? I didn't know that.
Yeah, it's a type of seabird. I have no idea how or why it's named that, and I have no idea.
Oh, okay.
But yeah, it's a type of seabird.
So we'll talk about the history of prion diseases in a second, and prions in a second. But so prion protein is this very important thing. You can have issues that randomly occur, which makes studying it hard, because we can never rule out when we're studying prion protein that it just did that. Like you just had a transcription error, you just had a typo, and then when you have that typo, you just die. But there's also infectious prion diseases. Did you guys ever see the Book of Eli?
Yes.
A long time ago.
Yeah, so when they were holding up their hands and looking for the shaking, remember that? Mm-hmm. Yeah, So that's supposed to be Kuru, which is a prion disease that was found in Papua New Guinea from people who were cannibalizing humans who were cannibalizing humans and consuming brains. So that's what that's a reference to.
Oh, cool. Or not cool, horrifying, but very interesting. Neat. Yeah, it's all neat. This is me a common reflection from me of that's cool and also horrifying. Should just be in the subtext of all of this.
Yeah. So when we get into the disease part of prions, just know that this is a real fuck you to foundational mammalian biology. These diseases, this is just unwinding your source code as a mammal. Without prion protein, you just die. Okay, so that's the whole thing. Okay, so let's just for a quick second talk about the history of prions and prion diseases, because I think they're super interesting. So they were first theorized and discovered in the 1920s by two German scientists, two German doctors, and I know what everyone's going to think right when I say that.
Yeah.
What did they do several years later? Mostly good things. So we have Hans Gerhard Creutzfeldt and Alphonse Maria Jakob. So that's where, if you've ever heard of CJD, CJD, that's where the C and the J come from. The D is disease. So those are the two guys, the two German doctors who figure out what prions are. Now, again, German guys in the 1920s, don't worry, neither of them went out to do Nazi stuff. One of them died before the war, and the other one helped people during the war. Was neither of them Nazis, so we don't have to worry about that. So prions weren't nailed down as the prion protein wasn't nailed down as a thing until the 1960s. And even then, like that definition of the field by John Stanley Griffin and Tavika Alper, like they couldn't like see the proteins. They could just they were just figuring it out. Prions don't get isolated until the 80s. Yep.
This is just really interesting how like they've theorized, they've cart before the horse all of this, which is just fascinating to me that you're able to do prions before figuring out what the heck a protein was. Like, oh, that's just it takes a lot of smarts that I don't think I got, but that's so
this
cool.
This prion disease has kind of first become an issue because of scrapie. You guys know what scrapie is?
Yep. It's a disease that affects sheep and maybe
goats?
Goats?
Sheep and goats. Sheep and goats.
Oh, okay.
And so it's a prion disease for sheep and goats, and that's where this becomes a thing. Because these sheep and goats die in a really weird way that's not like other diseases that impact sheep and goats. If you've ever worked with sheep and goats, you know sheep and goats are just like the most ridiculous animals that die for no reason sometimes. But prion diseases are very unique in how they present. Fun fact: in humans, we super under-detect prion diseases because it usually manifests like Alzheimer's or dementia or other brain diseases. So probably more than 1 in a million people die from prion diseases. But the only way to figure out if you have a prion disease that you died from is to cut the brain open and look at the brain. So it's probably it's probably more common in people, and we just are not doing a bad job. So, okay. So that's a little bit about prions and prion protein. Don't worry, listener, there's a lot more we're going to discuss. We do have some other plans for chronic wasting disease going forward as some side episodes, but we're just getting started with prion diseases. Okay, so prion diseases are an always fatal condition that are completely untreatable. If you get a prion disease, you die. That's just what happens. There's no ifs, ands, or buts. There's no outs. You're down. Your brain doesn't work. Lots of your body starts falling apart. You die. The biggest symptom, the telltale symptom for a prion disease, is your brain develops holes. So we'll talk about that in a second. But that's the primary symptom for prion diseases. Your brain just becomes full of holes and falls apart. So this type of disease is called the spongiform encephalopathy. So spongiform meaning sponge-like, encepha meaning head, and the disease. So spongiform encephalopathy. Let me just say that a few times. All right, guys. Let's say spongiform encephalopathy.
Spongiform encephalopathy.
Yeah, spongiform encephalopathy. So
encephalopathy.
The encephalopathy.
Yeah. So in animals,
get squishy brain. Get the squishy brain.
Yeah. I have that. Yeah. Wait, no.
No.
I feel like I have that some days.
Right.
God.
So brain should be pretty solid. Don't want spongy brain. Spongy brain equals bad.
Got it.
So in animals, in mammals, remember this is only a mammalian thing. This is a mammalian protein, so birds aren't going to be impacted by this, and neither are fish, neither are reptiles. This is mammals only. So in animals, the most common prion diseases that we see are scrapie, bovine spongiform encephalopathy, that is also called mad cow, but the real word for that is bovine spongiform encephalopathy, and chronic wasting disease. In humans, the most commonly seen is CJD. So that's going to be our Creutzfeldt-Jakob disease. So most of the time, that just comes up. That's just a problem. That's just a random mutation. You just randomly die from it. There is transmissible CJD. We'll talk about that in a little bit. And then there is also the bovine variation of CJD. So that was mad cow that killed people, and that was that killed most people in England. Almost 300 people died. Though interestingly, you can still find the variant of CJD from BSE in humans in the sewer system in England. So it's still killing people in England. It's just highly underdiagnosed.
I don't like that.
Yeah. Other things people may have heard of, again, are going to be cooler and sleeping sickness. Sleeping sickness is a genetic disease. Okay, so those are our most common prion diseases. There are other ones. We're going to put those all to the side. There was like exotic BSE. There was exotic spongiform encephalopathy, which happened in mammals in British zoos during the big mad cow or bovine spongiform encephalopathy outbreak in the 90s and 80s. But we'll put all that to the side. Okay, so those are our prion diseases. Most of the time, these prion diseases are not transmissible. Most of the time, they're just random mutations that occur. Sometimes they are transmissible, like scrapie, BSE, chronic waste disease, Kuru, and certain variations of CJD. We're going to talk about those. Those are called TSEs, or transmissible spongiform encephalopathies. Transmissible meaning you can get it from the environment or from something else. Okay. Now what do you guys see on the screen here? What's on our left?
On the left, you've got a normal slide of a brain.
What does it look like?
With, it looks, I mean, it kind of looks like a sponge too, because that's just what that shit looks like. But it's very solid colored for the most part.
Okay.
I was gonna say that's exactly what it looks like. There are very few holes in it. There's not a lot of white holes in it. There's different flavors of pigmentation, and then the one on the right is the one that has CWD. I'm guessing because it has all of the labels associated with it. But there's a lot of holes with white, where I'm assuming it's just straight through the
Yeah. So we're going to take a look at the animals that these came from. So we have so the left. So our solid. This is just like imagine a pink and purple slide. That's all it looks like with little circles in it of various colors. That's a healthy brain. So that came from a healthy mule deer, and then the one with the holes in it came from an unhealthy deer. So most of the time, these encephalopathies are mostly diagnosed when the animal is terminal and symptomatic. And so that's what we're seeing right now. So that is when an animal gets really skinny, their behavior is weird, they're drinking a lot, other things are happening because their brain is falling apart. So that's when it's mostly diagnosed. This is what it looks like, skinny deer. It's got like obvious issues going on. It's drooping, it's really skinny, it's wet because they're drinking a lot, because they're drooling a lot. So that's when it's mostly diagnosed. There's no way to diagnose CWD. You can diagnose CWD from brain slides. However, it's a really bad way to do it. There are better ways. We'll talk about how we diagnose them later. But let's finish talking about our TSEs. So those are transmissible spongiform encephalopathies. So some of these prion diseases can spread, just like we were talking about. Those are TSEs, transmissible spongiform encephalopathy. So we're going to just, for an example, we're going to use BSE and how that arose. So we believe that BSE arose because there was so in England in the 70s and 80s, what would happen is animals were fed rendered remains of other animals. And that worked. So you could feed, so you had sheep and like animals and the cows, and those were processed. So they go to the factory, they go to the slaughterhouse, and they cut up all the meat out of that, and anything that's left over, like the stomach, the brains, anything that wasn't eaten was ground up and then rendered, so it was cooked at pretty high temperatures historically, and then turned into a meal and then fed back to other animals. Well, then there were some corners got cut, and there were some changes made, and then they started to render them in a way that did not destroy the proteins. Because destroying a protein is pretty hard. And that's what we have to remember with prion diseases, is you're not killing an organism, you're trying to destroy a protein. So there were likely some sheep with scrapie that were killed and rendered and fed back to cows. Those cows then developed BSE from scrapie. Like I say likely because it's also possible that the cows just developed BSE on their own and then they were fed into they went through that whole process and were fed to other cows. So that's how BSE starts. Animals are eating diseased animals, and then they develop the disease themselves. And that's how we go to TSE, transmissible spongiform encephalopathy. A lot of the times, just like with a lot of diseases, it's not able to cross the species barrier. So, for example, scrapie has been found in sheep and goats as far back as the 15 and 1600s. However, we don't think that, again, all the caveats apply, it didn't jump the species barrier until the 1980s. So that's because that species area is pretty good, pretty high. So it jumps the species barrier into cows, and then cows, a lot of people eat a lot of beef, they're exposed to a lot of beef and a lot of products. Eventually it's able to jump its way into humans, and it ends up killing roughly 300 people. Just because that BSE was kind of unique. BSE was also able to jump into cats, so it killed some tigers and other cats in zoos, as well as some other zoo animals. Like it killed an American bison and some camels, just because of how that bone meal was treated and used in the British system. So that's our classic example of a TSE, starting in one animal, moving to another animal. So it was sheep to cows, cows to people. Killed at least 300 people, still found in England, though we don't really know how many people are still dying from it because it's under 7. It's just difficult to study. So that's the worst we've ever seen with a TSE. The impact on livestock was pretty immense. Most of the cows in the British Isles were killed. And then it also managed to make its way into the U.S. a little bit. And a bunch of cows here were killed. Export bans happened. If you donate blood, you can't donate blood or other organs if you spent time in England in the 80s and 90s, and that's because, again, people are worried about TSEs and about you potentially, if you potentially have some of these proteins in you, you could then give them to someone else and, spread that disease around. There's a very interesting case of a French person who got a TSE from medical equipment. So the equipment, so there was a patient before them who had a prion disease. It was undiagnosed. It appeared to be another brain injury, another brain disease. So they underwent brain surgery that was unsuccessful because they had a TSE that was improperly diagnosed. The person, so they underwent brain surgery. All the stuff, all the equipment was treated and cleaned, and then put away for 2 years. And then this next person who had to undergo brain surgery at this hospital, all that stuff came out, used on the new patient, went through all the hygiene processes, and that patient, the second patient, developed a prion disease from the first patient and died.
That's crazy.
Yeah, so these are...
I know that's how it works, but that's so...
Yeah, these are pretty robust issues. So...
Yeah, turns out it's pretty freaking hard to denature a protein.
Yeah, and that's also prions are why one of the reasons why most medical equipment just gets thrown out instead of reused. So pretty dangerous. I completely got sidetracked with that. Where was I going? We just finished talking about BSE. So
yeah, we were just covering BSE.
Yeah, so there's our BSE story. Now we do have you can still find BSE in the cattle in the U.S. There's been roughly at least 8 cases of BSE found from 2008 to 2025. However, these are all likely just random mutations that happen. There are millions of cows every year that are born in the U.S. and are raised, And so you're going to have some small amount of this happen. There has not been another case of transmissible BSE that's been found in the U.S., but there have been a couple of other cows that have had prion issues. Same, find this in the cattle supply around the world. A couple of cows every now and then. That's why we have regulation and all kinds of stuff around the slaughter and harvesting of animals. Okay.
For now.
Yeah. 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 That's one of the reasons USDA is super important.
Hey, USDA.
This diseased animals don't go to the food supply. Okay. So that's all of our most common prion stuff. It's scrapie and BSE. Well, now we come to the meat of the episode, which is chronic wasting disease. So chronic wasting disease is a TSE, so it's a transmissible spongiform encephalopathy found in cervids. It's 100% fatal to every animal it infects. We have to remember that. 100% fatal. Animals die usually within 2 years. There's a subset of animals that can survive, they can persist for longer, so they can live 3 to 5 years, just based on their genetics, but they all die. CWD was first discovered in the 1960s in Colorado. It was discovered at a farm where they were raising deer for some experiments, and it's likely that these deer were exposed to scrapie, and so they developed CWD from scrapie. That's what it's likely from. There's no way to know for sure.
So is it a So is that technically like a mutation, or is it just?
It's
okay.
I like a mutation. Usually, you're talking about genetics.
That's where I guess I'm getting confused.
Yeah, so it's not a mutation in that sense. It's just a protein that's the wrong shape that also happens to then make any other proteins that it touches take that shape.
So it's just one of those things, since there are that many proteins in this area with all the deer, that they were able to, it's just, a random chance after 10 trillion times folding a protein, it just started working. Is that kind of how it worked, I guess?
Yeah, it just kind of happened. Yeah. It's just kind of bad luck.
Okay.
Again, the species barrier is really high. We've had sheep in the U.S., domestic sheep and goats in the U.S. since the 1600s, and it didn't manage to jump into deer until the 1960s. So it's just bad luck. The species barrier is pretty high, but
I got that, but it's one of the things that I've always had a tough time figuring out. It's just it clearly jumped from one species to another at some point. It's just sheer volume of misshaped proteins that
probably
did it, and the answer seems to be yes.
Eventually, one of them hit in the right way.
Yeah.
Yeah.
But there's also no ruling out that this just randomly occurred in this deer herd in the 60s and then spread from there. However, when you look at, what happens to study this, what they do is they take CWD and you put it in a mouse model, and then you compare it in the mouse model to scrapie, and it looks very similar. So if you take it in the mouse model, it looks very similar to CWD, or scrapie looks very similar to scrapie. So that's why we think it probably came from scrapie, but just because of the nature of studying prion diseases, you never get to fully eliminate that just randomly developed. We're going to talk about later some examples in Sweden and Scandinavia where it looks like these are just random animals. These are animals dying from random mutations. It's unlikely that it's a transmissible spongiform encephalopathy, but that's a later thing. So just back pocket that one. Okay, so back to the origins of CWD. So it is first discovered in the 60s. People have been hunting and killing deer for a long time, really focusing on deer for a long time. So there is a conspiracy theory that CWD is endemic to North America, and it's just like a thing that has always happened. However, it's never been, and the behavior we see has never been recorded, until the 60s, until this deer herd. So I would say that that's completely unfounded. CWD is not natural. It is a new thing. The prion disease that we're dealing with is not natural. We're going to look at a map in a second here, and we'll see that it's definitely not natural. So it occurs in the 60s in these farmed deer. It escapes to wild deer in the western U.S. in the 1980s. That's the first time it escapes. So first found in Colorado, then it starts to spread into the western U.S., and then it jumps up into Canada, and then in the Midwest, then the Mid-Atlantic, and then a couple of points into the Southeast. Now, listener, you can't see this, because again, this is an audio medium, but what are you guys noticing on this map?
I mean, the far west and the southeast has, a few. It's basically it pops up in hot spots. So where it pops up, you get these big concentrations, and then you'll get new little hot spots popping up all elsewhere. But they're not all connected.
Discrete.
They're discrete
pockets, except for like the western United States, where it's basically that's just. There's clearly something going on along the spine of the Rocky Mountains where like it seems to be like there's a lot of elk and deer herds moving back and forth, but beyond that, like it seems to be they're all just discrete clumps that are happening.
Right. So listener, you can just look at U.S. Currently you can look at USGS CWD. You can find a map of CWD. Lord knows what's going to happen, with the various federal agencies, but just like everyone was saying, there's pockets of CWD that are not connected geographically. So we have like a pocket of CWD in Pennsylvania and Maryland that's pretty far away from Wisconsin and Michigan, where the nearest pocket is, and very far away from Colorado. So how do you guys think that CWD managed to get from Colorado to Pennsylvania?
I mean, the two ways are, transport of, farmed deer and products from them, or it's, hunters bringing trophies back. Or,
that was my
bring
bet. Was
whole carcasses essentially back, and then disposing of them just by chucking them in the woods, and then something else runs them over, and then it gets into the local populations.
Yeah. So the deer didn't walk here. That's the big thing I just want to point out, just to really nail down this conspiracy. The deer are not spreading this by walking all these places on their own. This is definitely human-aided. I would go so far as to say that it is mostly spread by captive cervids. So that would be captive cervids being elk, whitetails, blacktails, mules, sika, any kind of thing that you, like the high fence hunting things, those are all captive cervids. So this farming and raising, I'm using farming with scare quotes, and raising deer for products, I would say that's our driver of CWD. Because again, these deer are not walking from Colorado to Wisconsin. They're getting shipped there. There have been some really fantastic studies looking at the relationship of the spread of CWD on captive farms. So captive cervids are not regulated anywhere nearly like wild animals are. They're regulated as agricultural things. And most ag agencies don't really know how to handle deer because like these are like cow guys. They're like, I don't do deer.
At best, they're cow guys.
Yeah. So the other thing about ag agencies is they're always trying to promote additional production of that farmed resource. They don't care about wildlife.
They're not particularly interested in cracking down on on that.
Yeah. So we have these two distinct populations of cervids. We have wild cervids; these are free ranging cervids, and then we have captives. It's my firm belief that you get to either have captive cervids or wild cervids. You do not get to have both because of chronic wasting disease. So the current state of CWDs is found in 38 states and 5 countries. Those countries being South Korea. I guess it could technically be in North Korea. We don't know. South Korea, Canada, and then Scandinavia. That's where it's currently found. The Scandinavia being Norway, Sweden, and Finland. And obviously the U.S. So that's where it's currently found. How it got to Scandinavia, again, likely from hunter spreads, parts, stuff like that. So that's how it moved. Could have been reindeer that were also exported to Scandinavia from Canada. Not exactly sure on that one. Also, I haven't followed that the closest. We'll talk about the Norway stuff later. Okay.
Yeah, I want to say that the Scandinavian CWD has they think it has a different origin from, or
I know the moose one,
almost a different lineage, or at least that's one of the.
I know the moose one does. So that's another thing. Let's just mention that real quick. So CWD, there's a couple of strains. It's kind of you can almost
strain's the wrong word because that's like a virus and bacteria thing, but we don't have a better word for it.
Yeah, so there's at least 7 versions of it. Some folks are going to tell you there are 11. Um, we'll talk to some more prion scientists who can really dig into that. But just know, like, it's not all exactly the same. So like, there's the elk are a little bit different than deer than whitetails, and what's going on in Korea is a little bit different than the U.S. from a purely protein focus point. Regardless of which strain, again, scare quotes on the strain, regardless of which version of CW you have as an animal, you still get it and die. Again, this impacts all species of cervids. So moose get it and die, sika deer get it and die, whitetails get it and die, blacktails get it and die. Everybody gets it and dies. Guess what happens? There's no getting away from it. So how does it spread in the environment? Let's talk about that now. So we know it's this little protein. How does it spread? John, what do you think? How does it spread?
So this one I actually do know because while I don't know the science side of things, I've been in enough hunter education and kind of general DNR discussions and stuff like that. So basically anything that's like a saliva membrane of the deer, or really anything of the deer can carry it. Mm-hmm. So if it got shot, the blood can have it. If it's obviously the carcass can have it. If it was obviously taking a shit and urine and all that sort of stuff, like all that has it. They know like it's in like saliva, so like deer will like socialize and rub noses, so they think it passes through there. They think they know that prions are shed into the environment just kind of naturally, so that's where like all of the urine comes into play. You can come back and sample prions from that area where a deer went to the bathroom, and there'll be prions there, and a deer goes to sniff it, and now all of a sudden the prions get snorted up into a deer, and I guess they begin reproducing. They don't reproduce, but
no, fantastic. Yeah. No, they replicate it. Yeah, they replicate it. Fantastic.
They replicate. Yeah, they just do that. So, and if you have more deer, then you have more chances of these hits going off and spreading.
Yeah.
If you have a lot of like feeding stations, so that's like one of the big things is like if you've got a grain feeder, like here in Iowa, like people will set up a grain feeder thinking they're helping, and it's like, well, no, we have horrible prion diseases here, and they'll all eat, and if one has the disease, it'll pass to the other one, and you've just spread it everywhere.
Yeah, no, that's it. Yeah,
that's how it goes.
Yeah. So your vectors, the ones that people think of primarily, are going to be socializing among deer. So that's them, rubbing snouts, mating. Also, mother to young is another transmissible pathway.
Oh, yep.
So that's the classic one. Is any kind of deer fluid, so spit, urine, semen, all that kind of stuff. Any bodily fluid is a classic one. As we've studied chronic wasting disease, so this is where it starts to get scary. Remember, this is just a protein we're looking at. So the protein just needs to be anywhere in the environment. So other vectors include insects, so mosquitoes and ticks are another vector of CWD. So you have a mosquito that feeds on an infected deer. It can then feed on an uninfected deer and infect that deer. The other fun thing is that it builds up in the environment. So, infected deer they continue to produce chronic wasting disease, the infected protein, and they'll pee it out. That urine is then picked up by another plant. Deer comes along and eats the plant with the prion protein in it. Boom, now that other deer is infected with chronic wasting disease. It can also move in scavengers. So there's a study going on right now looking at ravens and crows and turkey vultures as the vectors. That's not a really easy study to do because ravens and crows are really tough to capture.
Yeah, they're too smart for their own good.
Yeah. So that's a tough study going on. But other things that you can culture chronic wasting disease out of hog poop, all kinds of stuff. You got to eat a deer or interact with a deer or interact with a deer's poop. And like worms, these are all vectors for CWD. All potential places to pick up CWD. Just like you said, another fun thing. This is scare quotes fun. Vector of CWD are deer licks. So where a deer will come. AJ? What's the scrape? A scrape.
A scrape. Yeah.
So there was a great study in Missouri where they looked at scrapes. So scrapes are this thing that whitetails do. There'll be a stick that hangs down off of a young tree. So it's kind of like Twitter for deer. They'll come up, they'll lick the stick, they'll rub on the young tree, they'll pee in their poop, and then everybody, every deer in the area comes up and checks on that. They see who's going on, who's been where. So the study in Missouri and infected counties found that 63% of scrapes had chronic wasting disease and could vector chronic wasting disease. So that's just imagine you open up Twitter and boom, now you got the Lyme virus. Now you got the killer, you got the killer disease. So that's how it would spread in the environment. When you think about managing CWD, there's kind of this 5% level. If you can keep chronic wasting disease below 5% of your population, that spread is, slower. And once you get past 5% in white-tailed deer, then it becomes, goes from like a logarithmic and a controllable, semi-controllable growth to an exponential spread. Once you get above 5%, then your population, just because of the buildup in the environment, it becomes almost unmanageable. And then you could head towards collapse because, you have buildup and you have every animal that gets, you have it building up the population. So does are infected, they're getting it to their young, it's everywhere. And so then you just have more and more animals getting infected until you get like southeastern Wisconsin, where like 75% of tested deer in some counties were infected.
Yeah, I was wondering if we were to get to the Wisconsin deer management strategy, we'll say. Definitely strategy in scare quotes there.
Yeah, that strategy is no management. But that's a future episode. We'll get a whole little sub-series on deer on CWD going, because it's a pretty scary thing. So what really concerns me about all of this is not just CWD in deer. Obviously they're important for a lot of ecological reasons as well as to humans, but also is the potential spillover. So that spillover being into non-humans. So any mammal in the environment is potentially exposed to chronic wasting disease, as well as livestock and humans. So that's what concerns me. Once chronic wasting disease, if it moves into a different mammal, then it's more likely to spread and to become problems for all other mammals. So that's the big scary thing for me. But let's just put that marker down. I'll come back to it. Okay?
Sounds good.
Okay, so where do we find CWD in deer? We find it in the entire animal.
Everywhere.
Yeah, the whole thing has it. So if you look at most agencies, they're going to tell you not to eat infected animals, that the whole thing just needs to be disposed of. People eat infected animals, or they don't test animals because they don't want to know. And they think, well, if I just don't eat the spine and the brain, where you find most of where you can find prion protein at its dentist, that they're not going to be infected. That's not true. Prion protein is in the whole animal. There's a fantastic study that was just done that looked at prion protein density and found that actually grilling was a fantastic way to increase the availability of prion protein in deer meat.
Huh. Is it just because you're shrinking the total surface?
No, you're heating it up, so you're just making it easier for your body to grab onto and to find.
Oh, okay. So if you can be like one of those weird people and just take a bite out of the heart once after you shoot it, that's actually lower risk than eating.
I wouldn't say that.
Yeah.
Let's not say that.
No, I think that's no.
I'm gonna
I
go down that route.
Actually,
yeah, remember,
this is not hunting advice.
yeah, this isn't food safety. Remember, deer contain lots of other diseases.
There's lots of other shit you can get from deer.
Yeah.
that cooking does kill. So keep cooking your food, people. We're not doing the
oh.
raw thing.
Yeah.
Oh, I thought you were one of those raw meat people.
Yeah.
Okay.
No. So how you kill CWD is not how you kill other diseases. You have to destroy the protein. So you have to heat it at 1,000 degrees for an hour, at 1,000 degrees. You have to bleach it in pretty strong bleach for 15 minutes, or you have to steam autoclave it. Those are the only ways to destroy the prion protein.
Now, our president once told me to drink bleach, so this bleaching solution idea, I think, could really work.
Yes.
I think that's honestly the best solution.
You just need to soak the entire animal in bleach for 15 minutes, and then you're good to go.
And I'm looking at autoclaves online. Like, they're anywhere from $40 to $5,000 or so. So you just You got to run that a couple of times. It should be
Yeah.
fine.
So how an autoclave works is it's steam and pressure that destroys stuff. So that's how they work. So if you don't do any of these things, we know that chronic wasting disease, the protein, can remain active and be cultured out of soil and plants at least 2 years, past when it's expelled from an animal. Now, the study that this came from ran out of money after 2 years, so I would bet that it could be found for much longer, but it was 2 years.
Our DNR guys, or the hunting managing guys, have said it's been basically for as long as they've been able to sample it for, they've found it there. So assume it's forever. Yeah. It may not actually be forever, but let's work with that. As something's going on.
Yeah. So that makes a lot of sense. I would say that it's functionally unkillable without bleach, heating to 1,000 degrees, autoclave, etc. That's where I would say it comes from. AJ, do you have other thoughts on just kind of the human risk from CWD?
I mean, you can't really overstate it. Just because it hasn't jumped yet doesn't mean it won't. Yeah. And the potential to get into the food supply is.
It's a bad pocket there. We'll come back to that.
Fucking terrifying.
Come back to that. We'll come back to that.
This is definitely one of those things where I learn about it and go, whoop, we're forgetting about it as soon as possible.
Yeah. And all the arguments of people, it's not that big of a deal. All of those arguments are rooted in fundamentally misunderstanding what CWD, what a prion disease actually is. Deer will eventually get immunity and all these other things that you hear. Because people don't understand that it's not a virus or a bacteria, so the immune system doesn't play into it. And it's not good stuff. I'm sad over here.
Yeah. So known vectors are animal to animal, deer farms, hunters moving carcasses and/or parts, carcasses on the landscape, rocks, soil, plants, also water, and then deer fluids. Other ones that are suspected and still being investigated are scavengers. Also, carnivores are a known one, and other wildlife. Also, fun fact on deer farms, one of the things you have to look out for chronic wasting disease on your deer farm are pets. If cats have access to the deer or the pens where the deer are kept, that is going to increase your risk level. Insects, ticks, all that kind of stuff. So these are all the things. These are all the places you could potentially get CWD from. Okay.
So basically everywhere.
Yeah. Cool. Okay, so let's talk about, I think, some of AJ's and my favorite thing. So testing for chronic wasting disease.
Yay.
So how we test for it in my state is you just take the whole head and you just drop it in, it looks like a big dumpster. You just...
Literally looks, it is a straight up dumpster.
Yeah, it's welded, locked together, so you can't just get in there and get after the head, but you just take a Sawzall or whatever and you just zip the deer head right off. Into the dumpster she goes. Biologists and techs come by every 2 days and collect up heads out of it. Also when you zip the head off, you put a tag on. You put your deer tag on it. It goes in the ear. So that tag and the whole head gets put on ice, shipped to the state testing lab, and then you find out if your deer is was positive or negative within 2 to 6 weeks. So that's how we test for it. It's free in areas that have CWD endemic or CWD is nearby. You can get testing done in other outside of those areas at a small fee, but it's also completely voluntary. There's no reason you have to do it. It's just recommended. I hunt deer. All of my deer are tested. Yeah, they do testing thoughts.
I mean that's essentially. Well, that will be how we do it once we get the coolers actually installed, and we haven't got quite that far yet. But to drop off locations, and then also I'm sure y'all's game commission does this opportunistic sampling from roadkill, that sort of thing too, and some of the CWD counts.
Oh yeah.
And all that.
Oh yeah. So the roadkill testing, that's actually, in my opinion, that's the best way to monitor for CWD in the landscape. So what happens in my state is the Game Commission has contractors that go around, and the contractors always try to beat up F-150s. They're always just driving not F-150s, shoot, what's the smaller version of the F-150?
Oh, a Ranger.
Yeah.
They're old ass Ranger.
Yeah,
man.
they always drive old beater Red Rangers, and the contractors go around and pick up any deer they see on the side of the road, throw it in the truck, and those all get sampled. They get paid per deer. It's gross. I would never want to pick these things up.
God, no.
But I mean, testing, roadkill deer is fantastic because you're getting a truly random sample, and you're getting it from year round or however whenever you're doing your sampling, and you're getting ideally from all ages. So with hunter collection, you tend to be sampling mostly males, though you should be sampling a lot of females as well. We talked about that already, but mostly older males because those are going to be the ones that have the biggest racks. There's also an issue testing older males in that a lot of hunters like to keep the antlers, and sometimes people haven't quite figured out how the Euro mount works. You just cut off the antlers, and then the rest of the head goes into the bin. So that can also be an issue. But when you do roadkill testing, you don't have any of these issues because you just pick up whatever's done on the side of the road. So you get all age strata, you get it from around the area. Fantastic way to sample. Also, it's pretty cheap. In my state, they'll also collect samples from any coal populations. So if you have, Valley Forge has coals that happens, so those animals get tested. We have an elk herd. All of the hunters are asked to volunteer samples. Most of the elk are tested because it's a very highly controlled hunt. So, we have all that. And private, on the private game ranches and stuff, testing is not required, and I have a lot more thoughts on private stuff that we'll get into. AJ, other ways do you guys test?
That's pretty much exactly it. I don't think we're at the point of doing any culls yet, and that's all I'm going to say about that.
Yeah, so we'll talk about managing seed in a little bit. So what happens after you get it? So what you're looking for in your testing is the biologists are usually looking to get the lymph nodes out or chunk of the brain because that's where you have the largest density prion protein. The lymph nodes can be pretty difficult to isolate if you don't know exactly what you're looking for. There was a fun study that came out a few years ago looking at vets trying to identify lymph nodes in deer, and they had about a 50-50 shot grabbing a lymph node or another gland or even part of the throat or random chunks of the brain. So that's why my state is we just want the whole head. We'll do that. Don't worry about it. But for the elk, they have a biologist there and a vet on site, and they just take the lymph nodes right out of the elk. It damages, if you want to do a whole head mount, it can damage that a little bit because they have to cut open part of the head to get at it. So you just do a European mount. It's fine. So then you take your brain slice or your lymph node, and you can either do an IHC, so immunohistochemistry, or an ELISA test, the enzyme-linked immunosorption assay. Or you do RT.
I have no idea what those words
are. They're just ways to just, regenerate, because they're just trying to make protein. So you just take that, and you're just blowing the proteins out that you're looking for. So the one that's coming up now that's really cool, it just got approved. It's been tested and used before, but it wasn't approved by, I can't remember if it's USDA or FDA or EPA. It needed one of those guys for approval, but it got approved now. RT Quick, real-time quaking induced conversion, also called shake and bake. So you just take your protein, you just take chunks of the tissue, you pipette it, and then you put it into this thing that just shakes it around at that temperature, and you just shake and bake, baby, and then you test that for prion disease, for misfolded prions. That's how you test for it. If you have a presence of these prions, then you have an infected deer. And again, in my state, if you have an infected deer, so in my state, they'll send you an email regardless of if you have an infected animal or not. If you have an infected animal, it needs to be disposed of properly, so that would be incinerating at a certified incinerator or put into a lined landfill. Those are the two ways that we want you to handle infected deer.
That's basically the same thing out
here. So that's how we want you to handle it, and that's because you're either destroying it in the incinerator or you're putting it in a place where it's not going to get back on the landscape. You don't want to dump it out back. If you dump that animal in the backyard, you now have CWD infected soil and plants behind your house, and then you have scavengers spreading it. This does mean, with the testing, it takes a little bit of time. It does mean, so for me, I shot a deer this year. It went to the processor because I didn't have enough room in my last place to hang and process a deer.
I was gonna say process an entire deer carcass.
Yeah.
Normal.
So to cut the meat out of it, and then grind it into sausage or burger, etc. I was living in an apartment last year, and I couldn't really hang a deer in the.
What? Come on! In your living room, your wife wasn't gonna let you do that? Crazy.
You're just hanging out well in the window. Yeah. Fine.
So it went to a deer butcher or processor, and I had him cut the head off. I dropped the head off, and then it was, I paid him. He cut it up, ground it into meat, and then all that. I got it back from him within 5 days, and it was in the freezer, sitting, waiting until I got my test results back, which was 5 weeks. So yeah, I mean, it does. Proper safety does mean you don't get to eat the deer right away, but it's fine. Man, it's fine to do. Some people don't like waiting for the process. Don't like waiting, but that's what you got to do.
Tough.
That or misfolded proteins in your brain.
Yeah. Or spongy brain.
Yeah.
Pick one.
Yeah, pick one.
Oh! One thing I forgot to mention is the prion seeding. So how prions start is you get, this is the scariest part, you get a small amount of prions, misfolded prions, and then the brain starts to copy based on those misfolded prions. And because you have a small amount, we can talk about how small that amount is in a second. So you have your small amount of misfolded prions as you need other prions, your brain's copy and paste random proteins, and we're going to use those going forward. As you copy and paste more and more misfolded prions, that leads to collapse because, so you get 3% of your prions are an issue. Eventually you're going to hit that 3%, and now all of a sudden 6% of your prions in your body are misfolded, then it's 9, then it's 10, then it's none of them working eventually. So that's the seeding concept. It leads to a collapse in the organism, which is why it takes deer 3 years to die, from infection. You don't go from a normal healthy deer to a deer that's slobbering, that's, hanging out by people in a year, because it just takes a long time for that seeding collapse to happen. Some biologists say that it takes about 5 micrograms, millionth of a gram. I think that's million, but yeah, micrograms, which is a millionth of a gram.
Yeah,
Some people will say it takes 5 micrograms. As we've gotten better and better at detecting prions, that seeding amount, the amount required to mess up your brain, keeps going down and down and down and down. So maybe that the amount of infected material you need to be exposed to get a prion disease is none. Was 1. You need to be exposed to, 1 misfolded protein, and eventually that's going to cascade and kill you. That's the fun thing. Okay, that was a lot about chronic wasting disease. How are we feeling so far, John?
Oh, horrified as usual. Nature continues to be just a bottomless well of just fascinating, cool things, but also things that are horrible beyond my comprehension. A little bit of protein, which is, however many molecules essentially stapled and folded and mushed together, can last in the soil at least 2 years and cause all this untold harm. Also, butterflies are really cool and pretty, and I like them. Man.
Yep,
it's a wide range.
Yeah.
It can encompass both of those.
both
of those
we can do
vibes.
both.
Yeah.
I'm not quite at animal fact level, but
okay. AJ, do you have other thoughts to add before we jump into the scary side of chronic wasting disease?
Oh, we haven't reached the
that's not the scary side?
We haven't talked about the scary side yet.
Yeah. No, I mean you've pretty well covered it. We're in the very early stages here, so I have a lot of thoughts that I will not be sharing on the podcast.
Yeah. Okay.
for professional reasons.
Yeah. So that is basic introductions to CBD. There's a lot more to talk about. We will be doing a future side series all about CBD, talking to CWD researchers and brain researchers and all that kind of stuff, because this is a really fun thing that scares the shit out of me. When I first got into CBD stuff, I didn't sleep well for like 3 months, but then eventually you just get used to it, and that's it. Okay, so things to remember about chronic wasting disease. It impacts all cervids. It is fatal in all cervids. It has not jumped the species barrier into humans that we know of. There are several ongoing long-term studies being, at the last I checked, they were being conducted. They could have ended now. They were being conducted by USDA, CDC, and USGS. And those...
Well, it's a good thing none of those agencies are, under any threat or anything, and don't have any problems there. So
yeah,
I'm sure it's.
It's also a good thing.
I'm sure they're still going.
That conspiracy theorists didn't try to make COVID out to be a prion disease.
Oh fuck! I forgot about that.
Yeah, that's been.
Wait, that's a thing.
Yeah, it's a thing. You don't. Yeah, that was really hard on the prion research community, which is a really tough community to be in to begin with. I went to a prion conference and those guys fucking drink.
I wonder why.
Yeah, that's a good drinking community there. Okay, so the species barrier is high. We've never found it in humans. I know someone's gonna think to themselves, well, what about that thing in Michigan? Yes, there were 2 hunters who hunted the same place that got CJD. However, they got them years. They were both detected within years apart, so it's unlikely that it came from hunted animals. It's just really bad luck. Remember, that is allowed to happen. Okay, here's where CWD starts to get scary. So in a number of animal models that we use to model human health, we have managed to infect them with chronic wasting disease. There's also a number of models. There are a number of animal models that we have not managed to infect with CJD. So, for example, squirrel monkeys can get chronic wasting disease, but macaques cannot get chronic wasting disease. Hamsters can get chronic wasting disease, as can a number of mice and mice models, but haven't found it in humans yet. So, the species barrier is high, but it is definitely crossable in a lab setting. And we know that prion diseases have jumped the species barrier, as we've seen with BSE. There is also this really fun thing. It's called the state of Florida. So in the state of Florida...
Talk about the state of Florida.
In the state of Florida, we have chronic wasting disease, and we also have several invasive populations of squirrel monkeys.
Oh, God.
Yes. Now, I will...
The dawning horrors are setting in, Kelvin. I don't like this. Monarch caterpillar photos. Hang on a second.
Now, I will say that those monkey populations are... So they have a couple other squirrel... They also have a couple other monkey populations that have escaped in Florida. They're mostly in the Keys and pretty far away from where the chronic wasting disease is at. But hey, man,
for now,
I said mostly, and also chronic wasting disease is pretty difficult to control, especially if you have a pretty big population, a pretty big captive cervid population, which Florida has.
And also, we'll say,
and they have hurricanes.
A laissez-faire approach to everything.
Biosecurity.
Yeah.
And also, you have this thing called hurricanes.
Yep. Oh, yeah.
Yeah. So, wouldn't it all shock me if we had eventually had some monkeys in Florida that ran into infected deer eventually? This is why invasive species are very important to control. The monkey populations in Florida should be eradicated for a number of other reasons. Oh, another fun fact about the monkey populations in Florida. I'm pretty sure it was the squirrel monkeys, but I think they have one of those populations that has Hep C at a pretty high rate. It's either the squirrel.
My head hurts, dude.
Yeah, so that's a real concern. It's either that or it's the rhesus. It's one of them. One of the monkey populations has a crazy Hep C, and you just shouldn't get near the dang things.
God.
Yeah, but they haven't been eliminated because people are like, oh, don't kill the monkeys.
Yeah, they're cute.
Yeah, shoot them dead. Okay, so that's one thing that is concerning. The big thing that concerns me is that we have chronic wasting disease in a free roaming animal that likes to eat corn and soybeans. We have lots of corn and soybeans in the environment. Not only do we humans eat that corn and soybeans, so does a lot of other animals, from livestock to wild things. So that's my
corn and so okay. Hang on, I've got a question for you. Could prions get shed in and onto an ear of corn? That corn gets processed into ethanol. I don't know how hot it has to be to process into ethanol, and then it gets put into my car, and then it gets sprayed out all over everywhere when I'm going to land a forestry job. Is that hot enough to
So the ethanol production is probably hot enough to destroy and eradicate.
Oh, thank God! Okay.
Well, that and the other stuff that you're exposed to that goes into ethanol production. There's a lot of nasty chemicals in that.
Oh, horrifying stuff! But who knows?
And the temperature inside the cylinder in your car is also going to be hot enough to denature.
And the pressure.
Awesome.
The prions.
Heat and pressure. Okay.
Heat and pressure.
But now
that's fine.
Let's go back to your example.
Thankfully, because I thought of that too.
Okay, good. I'm glad I'm not.
But let's stay with the dawning horror. So let's keep our ear of corn. Okay, so now what are our two most common uses of an ear of corn from Wisconsin?
Ethanol and
animal feed,
animal products.
Animal feed. So now we are at this time. We know we're doing this. We are feeding infected corn to all kinds of different livestock that are in stressful situations. Those stressful situations do open them up to increased likelihood of disease, and that we just know that that's very common with other diseases. So, these other animals include cervids. Feed corn is used to feed captive cervids.
Fatten cattle, yeah.
What's used to feed captive cervids as well as wild cervids, both legally and illegally in baiting. It's also feed corn is used to fatten cattle. So, in the nice, imagine we get, we know that this is happening. We have infected corn going to feedlots. It's also going into dairy. There's no reason like a dairy cow couldn't get in, fed infected corn, and now we have the dairy supplies infected.
I know there's a dairy right down the street from where we have at least 3 positives over the past 5 years of CWD in my area. So
if you drive around in the at dusk, you'll see deer hanging out in pastures. So it's very
hayfields,
very possible. Wild livestock is always getting exposed to this. So we're always testing that species barrier. That's one thing that scares me. The other thing is let's take our ear of corn now that's infected, and let's put it to the other really common use for corn in the United States, which is corn syrup. So now we got infected corn syrup. The corn syrup goes into
everything.
Mountain Dew. Now our Mountain Dew factory is infected, and that can go around the world.
Do you want some good news?
Yes, love some good news.
Please.
So that's what scares me.
At least according to IFAS, it looks like there are now no more squirrel monkeys in Florida.
Oh, hell yeah, dog.
Oh, shit.
The last population had only 1 monkey left in 2021, and then there's no update after
hell
that.
Yeah.
So
good.
They've got lots of other monkeys. Though.
Yeah. So the macaques are less of a concern. The other monkeys, I'm not super sure. I think the crab-eating monkeys may be a concern. I can't remember off the top of my head.
It looks like they got vervet monkeys. And I don't know.
Yeah.
But so maybe it's a little bit of good news there.
Yeah.
We'll take
invasive monkeys, never. Okay, so
not even once.
So that's my big concern is CWD getting into the food supply either through livestock or through infected plants, because we know infected plants do infect deer, and that's a good pathway. So that's my big concern. And then the other fun thing is let's say again in our corn example or a dairy example. That whole facility is now potentially infected, and anything coming out of that facility is potentially infected. Basically, every way we've tested to give model animals CWD, they've managed to get. So, that includes injecting CWD infected material straight into the brainstem of mice, feeding them meat that's infected, putting rolling steel ball bearings in infected material and putting that in their cages. That's a way to give model animals CWD. So, in an infected processing facility could infect the entire food system. Yeah. So there's the dawning horrors. How do we feel?
Only dawning.
How do we feel about the dawning horrors?
Depressed, anxious.
I'm so tired.
Yeah. Oh, and the other fun thing is, remember, it's difficult.
Stop saying fun, Calvin. It's horrifying shit, dude.
It is difficult to detect prion diseases, so it would take a, someone who's really on top of things to detect the first case, in a person. So it's difficult to detect. It is not impossible, and there are long-term studies that are ongoing that are monitoring areas with CWD, looking at prion diseases. And so far, those studies have not discovered any higher rates of prion diseases. There are ongoing studies looking at hunters, people who are known to have consumed infected meat. There are not increased rates of prion diseases, but that's not saying it can't happen. So
Yeah.
The species barrier. I want to just cap off the downing horrors section with the species barrier is crossable, but it is high, but it's crossable. So that's the scary thing. Okay, now let's go to the what can we do about it. So what can we do about chronic wasting disease?
Drink heavily.
Yes.
Kill more deer.
Yeah, we can do all of those things.
Yeah. Deer genocide.
Yeah, basically the only thing that we could do to, manage chronic wasting disease is to manage deer. At lower density, at lower deer densities, so it's deer per square mile, they are less likely to come in contact with each other to share chronic wasting disease, and it's less likely to accumulate in the environment. So we got to kill deer. Got to kill them.
Yep.
And then when we kill those,
and on the upside too, by committing deer genocide, the floristic richness, we'll say, so like all the spring ephemeral wildflowers, all the regular wildflowers also increase. Your tick population generally doesn't go any higher. It's technically spread by mice, but hey, it's probably pretty good that you kill more deer to try and control ticks. The amount of just other vegetation, like quantity and quality of the vegetation behind, also goes up. It's easier to grow trees. There's less car accidents. On the agricultural side, deer mow through tons of acres of agricultural crops. Personally, I don't care about it, but the rest of my state does. So that's always important for them. And there's probably some I'm missing as well. Oh, they spread invasive plants really easily. Like they just create tons of edge habitat, and
they move on their fur and in their
species I found.
They move on their fur and in
so
the invasives, not the chronic waste.
Yeah. So there's a lot of good that goes from what's the opposite of a keystone species, because I feel like that's deer at this point in the United States.
I mean, deer are a keystone species in that if you have too many of them, your ecosystem collapses.
Black hole of anyways, so that's how I view deer, is just whatever the inverse of like not a keystone species, but like one of those like net positive species, like wolves and beavers, like generally speaking, are overall net positives for an environment, but whatever the opposite of that is, it's deer.
I mean, they're just so overabundant almost everywhere in the U.S. that
makes a nightmare to exist with them, and everyone loves them. That's the part that annoys me the most.
Yep. Yeah. No, what else we can do? Yeah. Kill more deer. Don't have fucking captive farm deer. Don't do that. Don't transport whole carcasses, especially if you are harvesting it in a known CWD area and taking it to a non-CWD area. Just basic biosecurity stuff, guys.
Yeah. So killing deer and then testing those deer. That's the other thing we have to do.
Mm-hmm.
And then properly disposing of infected, meat and or carcasses. So not just dumping them in the backyard, but putting them in lined landfills or burning them in an incinerator. And then also cleaning our tools in between, kills and stuff like, in between kills and processing, as to not infect other things, as well as we're not going to feed and or bait deer at all. Be it legal or illegal, it's still getting them together. We're not feeding and or baiting deer. Another thing that's important is we got to have predators. Predators are very important. Not only do they disperse deer, they keep them apart, but help keep those populations lower. A fun thing about cougars are able to, in their stomach or somewhere in them, they are really good at neutralizing and denaturing the prion protein. So about 2% of the prion protein they destroy 98% of the prion protein that they consume from deer. So it's a really interesting study that was looking at that. I think it was done on zoo animals, but like,
do they know like the actual mechanism? Like, is it the stomach acid? Is it a particular enzyme? Is it
somewhere in the digestive system? Getting at that would require cutting the animal up after it had eaten. So it's something in the cat.
All right.
That research was pretty fresh as of 2 years ago, so I think that there's more work being done on that. But something in cougars is good at destroying prion protein. So if we just feed the next 2 or 3 generations of all cervids to cougar, that should about stop CWD.
Perfectly.
Except for, all of it that's already built up in the environment.
So the cougars, in addition to eating all cervids in North America, then we're
gonna need to feed them the top foot of soil in the entire state of Wisconsin
and the vegetation
and all the vegetation. Yeah. We got this.
Yeah, feed the carnivore all of the vegetation. Yep, got it.
Yeah.
Okay.
Yeah, this can
work.
We got
so there's a lot of cool things out there. Now we also know, having said that, that predators and cougars are not an end all be all. Like, again, we have to remember that CWD started in the Western United States and is still in the Western United States, and the West has relatively intact predator populations. You got grizzly bears. CWD is pretty common, or relatively common in Wyoming and Montana. And both, Wyoming and Montana have good wolf populations. They have good grizzly bear populations. They have good black bear population and pretty solid cougar populations. Yet CWD persists. So you're not getting it out with predators. Predators are cool and important, but they don't solve the problem. Wisconsin and Minnesota has CWD. It also has got great populations of wolves and black bears, and they're starting to get breeding cougars in the UP. Just having predators doesn't solve your CWD issue. So we can't just say, well, wolves solves this. Wolves do not solve this. They help, but they don't solve it.
I was gonna say, they definitely make our lives as land managers easier.
Yeah, and it's just cool to have wolves.
Well, yeah.
It's just cool to have these critters in the landscape because they deserve to be there. So,
yes,
let's not say that wolves solves this. Wolves does not solve it. That was good English. I nailed that. I'm proud of myself.
Yep. That's perfect.
Okay, and then the last thing, and I think the thing that we need to hit on the most, is captive cervids. Captive cervids are such a problem, and they're so underregulated and under-monitored. So in my state, if a deer in your farm dies, you do not have to report that death. I have seen deer stud books where a deer does not have one of its parents. So we don't, where did that animal come from? Doesn't have a mom or doesn't have a dad in some cases.
It's a divine miracle.
Yeah. So
that's how that works.
So I honestly...
You've heard of shrimp Jesus, but what about deer Jesus?
I'm very out on all things captive cervid, so I don't eat elk or deer or anything you can buy at the store because all that comes from captive cervids. I don't recommend that folks buy velvet supplements. I'm not here for that. Also, deer urine is an attractant that's used in hunting. Very against that. To get that, what they do is they put deer from their paddocks. They put them on concrete for a week, and then they spray down the concrete, and all of the stuff that gets from the spray down just goes into a.
Just be normal. Do you? Okay. I don't hunt enough. Do you have to do that? Can you just be normal? Yeah. Okay. That's what I thought, but I figured I'd ask.
You can in fact just be fucking normal.
Yeah.
Oh my god.
You could just bushwhack them like most people do. You could just sit in a tree or sit in a stand.
Just wait. Be patient.
Just go for a little walk. Hey, there's a deer. Ba-blam!
Do I even want to know what the deer velvet is for? Is it dick pills?
Yeah, it is actually, yeah.
Of course
Okay,
of course it is. What else would it be?
It actually,
It's always dick pills.
it actually is, yeah. It's also it's supposed to be like a testosterone supplement because deer antlers are the fastest growing bone, mammalian bone, and so there's a lot of I'm going to air quotes traditional medicine stuff around it. It's not, but it's whatever. What are you going to do? So don't eat that.
Great.
Yeah, don't do that. Don't eat captive deer products. I don't. It's way too risky for me. There's honestly a much longer conversation that we should have as a nation, but also as a podcast, which we will do later with experts about captive cervids. I would say we probably shouldn't even have captive cervids. This is probably a thing that doesn't need to happen. If you want to eat elk, go shoot an elk or know a friend who shoots elk. You know?
Yeah. I mean, we touched on it on the last series. Like, wildlife should be wildlife, and not be for sale.
Yeah. Sometimes there are some luxuries that you just don't get to have. If you want to see an axis deer, you got to go to its home habitat and see an axis deer. You should not see axis deer in Fort Lauderdale, Florida.
Sometimes you gotta spend a little money. Unfortunately, that means it puts it out of reach of some people, but it's not worth the cost at a grand scale to move a bunch of deer around and get God only knows what weird ass proteins in your system.
So those are things we can do. The other thing that we know that needs to happen is now I'm going to take us to we're going to look at some things that don't work for managing CWD. So ignoring CWD does not work. So Wisconsin has effectively done that. They managed CWD for a little bit. So it was first detected in 2003. For the first 4 years of detection, it was well managed. They were doing a lot of hunting on it, had additional tags issued in infected areas and were focused on it. Then that fell off after 2007, and now you have really high infection.
I wonder why.
Yeah, there's the Scott Walker's in there, the destruction.
Scott Walker.
Yeah, so
that fucker.
So then now you have super high, and so the total state infection rate is pretty high. And in some counties, it's 75% of tested deer. Whereas across the border in Illinois, when CWD first showed up, they increased harvest of does in infected areas and used sharpshooters and culling, when hunters weren't enough. Because hunters are okay at managing deer populations, but it is difficult to, get the culls and the population lowered as needed. Because, there's some number of deer that every hunter has that they're not going to shoot more deer after. For me, I'm not going to shoot any more than 3 deer a year because I don't have freezer space. I got freezer space for, 2. The third deer I can give away. A fourth deer, what am I going to do with that? It's a lot of work to shoot 4 deer just from a time perspective. The most deer I've shot in one day was 2. And I got really lucky on the second deer. In a couple of ways. I got lucky in that I saw it, and then lucky that it died at the foot of a friend of mine. I was like, hey, I'm going to tag it, and now it's your deer, because I already have this other deer to drag out. And, it was all day taking care of those 2 deer. So,
And also, the hunting population kind of at large is generally older, and there's not necessarily the same amount of replacement. I think we covered this in our wildlife management episode, but generally speaking, the replacement, there's not a 1 in, 1 out, or 2 in, 1 out. It's 2 out, 1 in maybe, and so that's playing havoc with both budgets, but also, hey, I need you to shoot 30 deer in this area, and you're 65, and it's cold outside in December in Iowa, and you're not going to do that. And I honestly, don't blame you, for not wanting to do that.
Yeah. The number one thing, we hear from people that we, farmers that we issue depredation permits to is they get out there and they do it maybe a couple times, and then they're kind of like, this fucking sucks. How do I hire somebody to do this?
Yeah.
Yeah.
Yeah. Well, and also hunters most hunters like to. I'm gonna say most because I hunt deer, but I hunt deer because I don't want to see deer. I want to see trees. But most hunters, hunt the animal because they're interested in the animal and they have a vested interest in the animal, staying around. And they believe that if we shoot all the does that the DNR wants me to shoot, I'm not going to have deer next year. Just because there are some myths out there about wildlife management. So there's also that to deal with. So that's why you need sharpshooters at a certain point. So you got to go down. So Illinois, they did targeted hunting. They brought in sharpshooters to cull animals out when hunting wasn't enough, and their CWD prevalence is around 1%. So that is success versus not success. So success can be had, but it takes effort. Other things that we know does not work. Ignoring CWD like we talked about. It's not going to solve itself. There is a theory going around right now that CWD will breed itself out. It will not. That's not how prion diseases work.
Prions work.
Yep.
It's not how proteins work.
Now, I will tell you that there is a subgroup of deer that are more resistant to, and this is deer and elk and some other cervids, they're more resistant to prion diseases. So meaning instead of dying in 2 years, they die in 3 to 5 years. Now, they don't survive, they just take longer to die.
And they pass along more
Right.
proteins. More proteins.
Yeah. They're just spreading it for longer.
So this is not a solution. The other thing that we know it's not. The other reason why we know you can't breed out of wild deer is chronic wasting disease is not the only pressure on deer population. So let's pretending that this genetics of resistance is heritable. We have first that resistance has to be heritable, and it also has to keep the deer alive, which it doesn't. Your resistance has to be heritable, and then for it to be bred into the population, CWD needs to be a major pressure on deer, and needs to be one of the driving pressures. We know CWD is not the driving pressure on deer. The driving pressure are people with guns and bows, predators, and habitat. Those are driving pressures. So it's unlikely that you're going to be able to breed in this resistance. Again, air quotes resistance, because you're just taking longer.
Actually, resistance.
Yeah. I know it is theoretically possible in captive herds that you could breed for this resistance, but again, it's resistance. They die anyways. Okay, those are things that don't work. Another thing that does not work: fire. You will not burn this off of the landscape. I know that we all like fire. Fire is a lot of fun. However, prescribed fires do not reach 1,000 degrees Fahrenheit for an hour. They get a couple hundred degrees for a minute to seconds to minutes, and then they're gone. This is not the same as putting entire landscapes into an incinerator. If you are burning at 1,000 degrees for an hour, you have a lot of other problems. You may solve your chronic wasting disease problem, but now we run into the problem of there is no vegetation left. Oh my God, a volcano has impacted all of this area. So we're not burning it up either. So those are things that will not solve it. Also, conspiracy theories do not help. It just takes, honest to God, focused management and effort.
It takes long-term focus, funded management. It does not. This is not something that will be solved in an instant. This is something that will take time and energy, and like with everything in natural resources, it'll take something that takes longer than 30 seconds or one election cycle to get done.
Yep.
Turns out those public agencies that some of us work for are actually important. Who knew?
Wild, that.
Crazy.
Yeah. Okay, so that's been our introduction to CWD. There are a lot more conversations and questions that we can get into that we have not today. We have a little side series that we're thinking about doing that we'll be working on in the future. We got to get better at podcasting first. So that's been our intro to CWD. Do we have... Oh, I almost forgot. There's a whole international side to CWD that we have lightly discussed. That will be in our side series on CWD. We're going to just put that whole thing in the side series.
Yeah. That'll be too much.
Yeah. Creatures impacted in the side series or in the Scandinavian are reindeer and moose. But that's a teaser for the side series. Now, before we sign off, gentlemen, do we have any additional questions and or thoughts about chronic wasting disease, or as the researchers call it, chronic?
No, more just not less daunting horror and just active horror.
Yeah, that about.
All right, we've hit the chronic pretty hard.
Yes, we have.
Okay, so this has been another episode of Seed Pod. This is Calvin signing out. I think that our next episode is going to be feral horses.
Maybe.
I think maybe.
Yeah.
Hopefully.
We'll offline about that.
Or who knows? Maybe it'll be a surprise. We'll see.
We'll offline.
See everybody later.
Bye.
Bye.